My name is Brandon Mayfield, and this is my project on FSC 435 Policies and Procedures. This is a review of detectability, visualization, and DNA analysis of bloodstains after repainting the walls. So the purpose of this experiment was to duplicate a crime that was committed in Germany, where a couple murdered a landlord with an axe. Upon investigation, these policemen discovered that their walls were all dingy except for one. One was freshly painted, which raised some eyebrows, and they got pretty suspicious. So this group of researchers decided that they wanted to determine whether or not freshly painted walls could cover up bloodstains or not, and what happens over time to these bloodstains. For luminol, don't know why it's not coming up. Sorry. So luminol is a chemical that is used by forensic scientists to identify trace amounts of human blood that's left at crime scenes. It's a chemical that reacts and glows blue with the proper oxidizing agent. When it's mixed with hemoglobin and human blood, luminol will shine, as in this photo shows. This is human blood, and a luminol test is being done right now in this photo. There were two types of walls for this experiment. There was setup A, which had the fine-grained lime plaster, and B had the sheetrock walls that were layered in wallpaper. And for the blood supply, a physician with a healthy blood count and a body temperature of 36.6 degrees Celsius donated his blood to be used in the experiment to limit the variances in data. His blood was used within 1 minute of being taken out of his body. So with the procedure, he took 2 rows of 5 handprints, and with equal amounts of blood on them, 2 milliliters per hand, mixed together and placed, and then they put 2 more milliliters of blood and placed, and they did that for all 2 rows, 5 columns. The top row was right-handed, the bottom row was left-handed handprints. The top row is labeled A1, B1, C1, D1, and E1. Bottom row, A2, B2, C2, D2, and E2. After the 7-day drying period was over with, A1 through E1 was painted 3 times, A2 through E2 were painted 4 times, every 12 hours. And after 9 days following the last application of paint, there was 5 milliliters of Lumicine Ultra that were sprayed for 30 seconds from 100 centimeters away onto the wall. This shows the columns and the rows of A1 through E1 and A through E2. And it's the top row that only had 3 rows of paint, the bottom row had 4 rows of paint. As you can see from this figure, there was 5 at the top and 5 at the bottom. This is a sample handprint to distinguish the 2 rows. So these figures represent the bloodstains before there was any paint added. These had to dry, and then coats of paint were added, so where you couldn't see the paint, the hand anymore. You could only see paint. And they were not clearly visible to the eye, which allowed them to do a luminol test and get some data from it. This is the second setup, where they did one set of handprints, 10 large splatters of blood, and 20 small splatters of blood. And this is before 6 coats were added to the wall, as shown in this figure right here. And as you can see it pretty well, the large ones especially, but right here, they're not really visible to the human eye anymore. And so for that's for setup B, that second set of pictures. that was for wallpaper. And so all of the stuff was left for 7 days, and that's when they added those coats, which were created these right here. And this is a chart that shows when the paint was applied. It shows 8 a.m. was not administered for the top row at all. Every third day, that was the case. But every day, the second row, the bottom row, got 4 treatments every 12 hours for 2 days at a time, totaling 10 days. And on the 11th day, luminol was sprayed, and on the 12th day, repeat luminol spray with water, distilled water, and that helps the luminol detect the blood on the wall. Without it, you wouldn't see it as well. These images indicate that the intensity of the chemiluminescence of the bloodstains decrease as the drying time of paint increases. The top row of palm stains as 3 coats of paint versus the bottom row that has 4 coats. You can see at the top row there was still a pretty heavy amount of blood stain that you can see through the luminol. You can only see just a little bit on the bottom row, but it is still visible to a degree. And that was after 1 day of drying time. The visibility of the contact blood stain decreased as the time allowed for the paint to dry increased. This made it nearly impossible to visualize after 8 days of drying time. So far on the data we've collected visually that, well, the more paint you add, the less you can see the bloodstains on the wall. However, the bloodstains are still there. It just takes a little more prep to get them. So the bloodstains A2 through E2 with the 4 coats, that was the significant reduction in bloodstain visibility. It was found that the bloodstain was only chemiluminescent using luminol for the last 24 hours of drying time. And let's see. So this also showed that the wall surfaces were pre-treated, the ones that were pre-treated with water, distilled water, those in A1 through E1 and A2 through E2 were more likely to result in visibility given the circumstances of water. They said that the more the time gives to dry, that the more likely it is that you won't be able to see the blood stain. So here we have a graph that shows the large and small blood droplets decreasing over time to where the large droplets, which you can visibly see, are almost invisible, but they're still visible. These ones were slightly visible, the small droplets, but they became nearly invisible, almost to where you can't really see them at all. And after 6 consecutive applications, you see the next 4, 5, and 6, all of the prints, bloodstains, everything became nearly invisible. But this is before the luminol and before the distilled water. The macroscopic visibility for the human to see the bloodstains with the naked eye decreased as the time went on. A total of 6 coats were applied, and they ended up concealing the bloodstains completely. Testing the walls with 6 coats of paint with luminol uncovered only small pecks of chemiluminescence that indicated artifact. So this shows after it was distilled in water for 15 minutes. You can see the significance in the luminol. It made it very visible compared to this this on the left without luminol and without water. And that makes it easier for the scientists to really get good data as far as visualizing it. If the wall surface was pre-treated with water, the 42 out of 50 droplets, or 84%, were positive for chemiluminescence. But when you only allow it to dry for 2 days, 100% of blood stains were detectable with chemiluminescence. But if you gave it 7 days to dry, you would only have 80% effectiveness for chemiluminescence. The increased amount, one thing we can gather from this so far is the increased amounts of drying time equals decreased amount of visibility of blood stains. And it can only detect about 70% of blood stains using chemiluminescence, but that's after allowing the paint to dry for 240 days. Here you can see that as the days go by, this is 2, 7, and 240, the negative test increase significantly, while the positive test decrease and then increase back to 240. And this is for DNA over a time period. The results of the descriptive statistics of times, depending on DNA results, you got 2, 7, and 240, and you can see the numbers significantly change, although there is a slight change after such a long period, being 240 days. It just increases slightly. This is the difference in DNA depending on the time that has been elapsed between paint coats and DNA, DNA sampling, So for the DNA analysis, 37 out of 50 samples, or 74% of the bloodstain samples used, produced positive results. Those DNA positive samples underwent DNA typing, and all those samples generated full short tandem repeat profiles. The rate of success of DNA positivity in relation to the amount of time the paint had to dry indicates further that the longer the paint has to dry, the less likely there would be a chance of conducting proper DNA analysis. In conclusion, the results of the study reinforce the usage of a regular crime scene approach of using luminol to test the walls and objects of a crime scene that appear to have a fresh coat of paint or are newly renovated. It would be valuable to consider that the objects that contain a high absorptive capacity would likely benefit from the pretreatment used in this study. The relationship between experiment, policies and procedures, quality assurance and control, safety, and ethics learned in this class, they really taught me a lot when it comes to understanding how to analyze data that comes through in an effective, safe, and ethical manner. And this concludes my research project with my resources. Thank you guys for listening.